Okay, your next topic is microsporogenesis. So in the last class we have seen that microsporangium, okay, which is the anther. So inside the anther the microspores are formed. Microspores means it is the another name for pollen grain. Remember that. Don't get confused, microspore and pollen grain. So the pollen grains are also known as microspore, which are formed in microsporangium. That means inside the anther, in the pollen sac. Okay. Now microsporogenesis means process of formation. So the process of formation of microspore or pollen grain from microspore mother cell, okay, which is also known as pollen mother cell in microsporangium, is known as microsporogenesis. So remember the definition: process of formation of microspore or pollen grain from microspore mother cell or pollen mother cell in microsporangium, that is anther, is known as microsporogenesis. If the site is asked what is the site of microsporogenesis, then it is microsporangium. Now what is the process of formation? This is a very important question, very important question, and it usually comes for 5 marks. So the question comes as write the development or describe the development of microspore from microspore mother cell. Okay, so the initial tissue from where the pollen grains will form is the sporogenous tissue. So if you remember, the sporogenous tissue is the homogeneous type of tissue which is present inside innermost tissue of the microsporangium when it is in immature condition, that is enclosed by the tapetum. Okay, so sporogenous tissue is present inside the microsporangium only, which is diploid tissue. Okay, so diploid tissue which undergoes first, initially it undergoes mitosis to form microspore mother cell or pollen mother cell. Now this pollen mother cell, PMC or MMC, further undergoes meiosis, which is the reductional division to form pollen tetrads. Okay, why it is called tetrads? Because see, the 4 pollen grains on right side I have drawn on these 4 pollen grains, they remain together, they are attached together, and this is enclosed by a callus layer. Okay, this is enclosed by a callus layer which is also synthesized by the tapetum only. This is a layer which encloses the 4 pollen grains together. Now on maturity, these pollen tetrads gets detached from each other. Okay, they gets detached from each other and dissociate from each other and gets arranged at the 4 corners of the microsporangium. See, on the next step I have written gets arranged at the 4 corners of microsporangium, and in this similar way only this that means from 1 PMC or 1 MMC 4 microspores are formed. Similarly, there are many PMC and MMC inside the microsporangium. In this way, hundreds, thousands of microspore or pollen grains are formed inside the microsporangium, which on maturity the these layers, that means the epidermis, endothecium, middle layer, they dehiscence. Okay, they undergo dehiscence and releases the pollen grains outside. So you can see in hibiscus the powdery like substance, yellow color powdery like substance present on the surface of stigma or present on the surface of the anther. So they by the process of dehiscence, they just come out from the microsporangium or they undergo self pollination. That's why you can see these yellow color pollen grains, powdery like substances on the surface of the female organ or on the anther only. Now this is up to pollen grain. So remember one thing, pollen grain is just the carrier of the male gamete only. It is not the cell or the structure that helps in that is involved in fertilization. It is just a carrier. That means pollen grains carries the male gamete from the male part to the female part, but it is never involved in fertilization. That means there needs to be developed inside the pollen grain, there needs to be developed developed male gamete. Okay, now we will see how male gametes are developed inside the pollen grains. So see, next step from pollen grain. So pollen grain is a haploid structure, therefore I have written N, but sporogenous MMC, till you will get mitosis, it is twice N. Okay, but once it will undergo reductional division, it will be N structure. Now this pollen grain further it will undergo mitosis to form 2 unequal cells. One is the larger vegetative cell, another is the smaller generative cell. This is 2 unequal cell. Vegetative cell is generally larger, but generative cell is the smaller one, and generative cell floats on the cytoplasm of the vegetative cell. That's since vegetative cell is the larger one. Now most of the angiosperms, that means 60%, you can refer to the notes there, it is written, most of the angiosperms, that means 60% of flowering plants release their pollen grains at this 2-cell stage. Okay, so at this 2-cell stage only they will release from their own body means from the male organ, and after falling on the female reproductive organ, the further development takes place. Actually here, if you will notice, there is no formation of male gamete, but our main aim is the formation of male gamete. Okay, so this generative cell needs to be further undergo division. So again, if you will see in the next step, after 2-cell stage, this generative cell further undergoes mitosis and results in the formation of 2 male gametes. Therefore, there it is 3-cell. So hence 3-cell stage. That means 40% angiosperms release their pollen grains in 3-cell stage and 60% in 2-cell. That means pre-pollination, if you will see, before pollination only, it is a 2-cell stage, which after pollination they will undergo further mitosis to form 2 male gametes. But in the last stage, in the pre-pollination event, that means in pre-pollination, before pollination only, in the anther of the male part or the male reproductive organ or the male flower, wherever it is present, there only it will undergo the mitosis of what? Mitosis of the generative cell to form the 2 male gametes. Okay, so this part you just remember. If you have any doubt, you can ask me. You can call me and ask. So, but this should be clear. Okay, because this is an important question. This part is very important. So again, I will say sporogenous tissue undergoes mitosis to form MMC or PMC. This MMC or PMC further undergoes meiosis to form pollen tetrads. They are attached together, enclosed by a layer which is known as callose layer, and this layer is synthesized by the tapetum only. Okay, that nutritive tissue. Now on maturity, these pollen tetrads gets detached, dissociate from each other and arrange themselves at the 4 corners of the microsporangium. So till this, microsporogenesis is complete. Now next, the microspore undergoes mitosis or the equational division to form 2 unequal cells. One is the larger vegetative cell, another is the smaller generative cell. Now 60% of angiosperms release their pollen grains at 2-cell stage. Okay, at 2-cell stage, that is one larger vegetative, one smaller generative. Whereas in another 40% angiosperms, the generative cell further undergoes mitosis to form one larger vegetative cell and 2 male gametes. Okay, therefore they release their pollen grains at 3-cell stage. So remember these steps and always present one flowchart. This needs to be written in para. Okay, in paragraph you have to write, and also along with that paragraph you can give this flowchart. So it will be better. Okay, so presentation will be better. So this is all about microsporogenesis. Now next we will see about the structure of pollen grain or the structure of microspore. Okay, so you can see the pollen grain consists of 2 layers. Okay, inner and the outer layer. The inner layer is smooth generally, but the outer layer is rough in structure and it is hard. Okay, so the inner layer is known as intine and the outer layer is known as exine. Remember that, intine and exine. Now this exine consists of a chemical. It is made up of a chemical which is known as sporopollenin. See, the word spore from microspore, pollenin from pollen grain, the sporopollenin. The name of the chemical is sporopollenin. Remember, this is very important question, very important, because this chemicals, sporopollenin, presence of these chemicals give resistance to the pollen grain. Okay, give resistance to the pollen grain, and because of this chemical, it cannot be degraded by any type of acid, enzymes, or you can say high temperature, pressure. So this sporopollenin gives resistance to the pollen grain in many ways. And due to this presence, due to the sporopollenin, the pollen grains can be kept for many years. Okay, they can be kept for many years as fossils. So these questions comes as, what is the property for which pollen grain can be kept as fossils? Okay, what is that property? So remember, due to the presence of the chemical sporopollenin, and on which layer if it is on the exine. Okay, but the thing is that the sporopollenin is not present throughout the exine layer. It is not present throughout the exine layer. In some point, in some part, this sporopollenin is absent. Okay, the sporopollenin is absent. The part where sporopollenin is absent, that part we call it as germ pore. Okay, the part where sporopollenin is absent, in that part germ pore is present, and through the germ pore the pollen tube protrudes out. You can see on the right side I have drawn one figure. From the intine layer, inside layer, the pollen tube comes out. Okay, and what is that part from which the pollen tube comes out? It is the germ pore. Remember, so function of germ pore, if it is asked, the protrusion of pollen tube. And sporopollenin, remember that, very important question. And in the inner layer is the intine. Intine consists made up of protein and carbohydrates. So together the protein, pectocellulose, we can call it as pectocellulose, or sometimes it is also written as pectin and cellulose. Okay, pectin and cellulose, and hence together we can call it as pectocellulose. So this is structure of the structure of the pollen grain. Okay, and in some flowers which are majorly insect pollinated, okay, insect pollinated, the exine is covered by a yellowish sticky oily layer, okay, which is called as pollen kit. As I have discussed, the pollen kit which is secreted by tapetum layer. The other functions, the main function of tapetum is to provide the nutrition to the developing pollen grain. Other than that, the pollen kit, okay, pollen kit means that sticky oily layer which attracts, or which attracts means which helps the which helps the flower to or helps the insect to attach the pollen grains on its body. Okay, this is known as pollen kit. So this is all about the pollen grain, structure of pollen grain, exine and intine. And another term you have to remember about pollen viability. Okay, viability means what? See, now there is a time period for which the pollen grain retain its ability to germinate. Okay, after falling on the female part, that means it has some time period, particular time period, that within this time period if they are able to germinate, then it's okay, otherwise they are unable to germinate. Okay, now the time period for which the pollen grains retain the ability, okay, they retain the ability to germinate, it's known as its pollen viability. That means after falling on the female stigma, the protrusion of pollen tube, or if they fall on two cell stage, further mitosis division, so there is a particular time period within which they have to complete these stages. So that particular time period till which they retain its ability, we call it as germination, till it's retaining the ability to germinate after falling on the female reproductive part, is known as its pollen viability. But it is different for different plants. Okay, some plants retain its viability for months, but some plants has only for 2 to 3 or you can say for very short period of time. That means within 30 minutes they lost their viability within 30 minutes. Okay, now pollen viability for some plants like rice, or you can say wheat, they retain their ability only for 30 minutes. After that they lost their viability. But whereas families of plants, Solanaceae, Leguminosae, Liliaceae, they retain their ability, they retain their viability for months. Okay, so that is about pollen viability. Remain viability means the time period for which they are able to germinate after falling on female organ. Now it depends upon the environmental condition also, like temperature, humidity. Okay, so they depends on these factors also. So this is about the structure of pollen grain.